Welcome. This is the second lecture of Engineering 443-543, Computational Modeling Methods, Virtual Tissues. I need to remind people that the class is live streamed and archived and publicly available, so please be aware of that. I know Zoom rewards you when you come in of that, but I want to make sure that that's clear to people as well. I'll ask people, feel free to break in and say things. Feel free also to use the chat, but please remember to mute your mics when you're not speaking so that we can avoid background noise. Giuliano is an unofficial assistant for this class. He's a graduate student in ISC. He's very experienced in CompuCell. He helps teach the summer version of this course, and he's been a teaching assistant for this course before. And he's an expert. He's also an expert at nanoHUB. So everything about how to upload projects to nanoHUB will go to him. He's the expert. And if there are bugs in the nanoHUB deployments, which people seem to have found, he's the one to go to in terms of getting the nanoHUB issues resolved. So, last time we introduced the basic theme of this course, which is going to be how do you model organismal development, homeostasis, and disease. In particular, how do we understand the interplay between physical mechanisms and biochemical signaling. And we're going to do that in this class using computational models, which we tend to call virtual tissues, although they also have other names. The virtual tissue name was developed by our colleagues at the Environmental Protection Agency. I think it's as good as any. And typically in the models that we're going to build, we're going to have two or more scales. We're going to have a cell scale at which we have cells represented as spatial objects that behave, that interact. We'll often have signals and background information at larger scales in the tissue. And later on in the class, we'll find that we can actually go quite far with the cells having essentially static behaviors. But later on in the class, we're going to introduce the idea of gene regulatory networks and signaling networks within cells so that the cells can have more intelligent response to their environment. There are other modeling methodologies that go to smaller scales and larger scales. We're working at this mesoscopic scale of multicellular organization. And then last time we also introduced a bunch of examples. Some of these examples actually we skipped in the interest of time, but one of the things that this kind of modeling does is produce movies, and these are sample movies of different kinds of biological situations one could simulate, from a detailed model of a single cell crawling on a substrate to a very abstract model of somatic evolution in cancer, to more spatially detailed and realistic models of here tumor growing surrounded by blood vessels, here of the formation of a cyst in the kidneys, here of blood vessel invasion in the retina, or these models of viral infection in a patch of lung tissue, or on the top right, a fluid flow in the liver. This again is mainly to illustrate the variety of models that one can build, all built with the same set of basic tools. Today, we're going to talk a little bit. I'm going to ask people to introduce themselves very briefly, maybe talk for a minute or two about their project ideas if they had them from last week. That was one of the homework problems. That was homework 4. I realized trying to get people to think about projects at the very beginning of the class may be challenging, but we could always talk about these ideas. I'd like to get people's ideas a little bit on homework problems 2 and 3, just to get some feedback on that. Then we'll talk a little bit about how to upload and download simulations to nanoHUB and run them. And then we will start talking about how CompuCell works and try to do some basic simulations having to do with how cells are defined and how you do very simple things like change the size of a cell, the shape of a cell, whether the cells stick to each other or not, and so on. And I hope by the end of the class, we'll also do a little bit of programming in terms of how you put up scientific plots in simulations. One of the things that you need to do typically in these simulations is measure something like how big the cells are, how many cells do you have. And so I want to go over quickly the code, 3 or 4 lines of code it takes to do those kinds of plots. So to begin with, why don't we go around the room and have people introduce yourself briefly, what department you're in, whether you're a graduate student, undergraduate, what your interests are, and if you, I know some people didn't get the homework assignment in a timely fashion because of the problems last week, but if you have a slide or just want to talk for a minute about your ideas for possible projects, that would be fine now as well. So maybe we'll start with Joel, since you're at least on my screen at the top left. Why don't we start with you?
Sure. I am trying to find the file here to see if I can share with you guys. I don't know if it's, well, it's not problematic to show this part of the homework, I guess. So let me share my screen just a second. Okay. Yeah, so my name is Joe Vanin, as you guys can see. I'm working close with Glazier. I've been admitted on this spring, so everything is very new for me. I'm a PhD student. And I'm working mostly with modeling right now with James. And one of the interesting projects that I found was representing necrotizing enterocolitis. I don't know the term in English, sorry. But this paper is a already done model by a different team, and the idea is to represent the same model but using CC3D. So it's a very common disease, especially very common is 25% of all premature babies end up contracting this disease. And it's important for us to understand a little bit better what is the cause behind it. The team had a hypothesis around this that was just the OS, I forgot the term, just give me a second here. Yeah, oxidative stress that was not being cleared from the system in a manner, in a timely manner. And that was their hypothesis of the problem of creating necrosis. But after their test with their models, they noticed that there is more things involved. It was not only the clear of the OS that was causing necrosis. It was possible to have a lot of OS in the system and necrosis was not started. So they didn't dive in the other discoveries that they did, but is relate to the mucus barrier and the TLR4 activation. So that's something that I am also willing to show better if we can make it this model on CC3D and try to go further on this second hypothesis generated after their paper.
Okay, great. I should say that I know some of the people in the room are auditors, so you may not be doing the homework, so you may. But even if you're auditing, if you're willing to just briefly introduce yourself, that would be very helpful so people know what your expertise is. Again, I think one of the great resources of this class traditionally has been its diversity. And so knowing a little bit about people's backgrounds and interests can be very helpful. Maybe to move on to Alex.
Hello, I'm Alex. I'm a third-year PhD in the engineering department, but my focus is mostly operating systems and cloud engineering, not really bio stuff. That kind of impacts my view of the project because my bread and butter is like the C++, Python, lots of programming. So I'd be more interested in like doing some kind of plugin or whatever for CC3D. But then like I do know a little, I mean, I do know biology well enough, but not this kind of detail. But I do know, at least I'm aware of the blood cells invading the retina. So it's also an interesting option as well.
So one of the things we could probably do is hook you up with TJ and see if there are projects, software projects that are appropriate. The alternative, I don't require people, but I strongly encourage people to work on projects as teams. Another reason I'd like people to start talking about possible projects early. And in that case, potentially, if there's somebody who's more a biological expert, teaming up with somebody who's very good at coding could be a very functional team. And so we'd be fine to come up with a software project. Certainly people have done that very successfully in the past. But it's also potentially, if you're interested, possible that there might be somebody who would appreciate that coding experience. Definitely C++ extensions of CompuCell and other packages are valuable. And it's actually not that often we get people who want to do that. So I don't want to discourage you. TJ often comes to these classes, but he's tied up today. But we could hook you up with him individually. Great, thank you. Connor.
Hi, everyone. I'm Connor. I'm a first-year PhD student in the cognitive science department. So originally, before coming here, I did a little bit of work in biology in terms of behavioral observation in skate embryos. Now here, I'm mostly doing simulations with genetic algorithms for different types of kind of simplified systems to look at the evolution of different types of plasticity and adaptivity. It's usually, though, in that domain at a more abstract level than what happens here, but I think that there's room for interesting collaborations where that amount of detail is included. In my homework, I listed down kind of two potential paths. One, which is a goal I would like to try to get to at some point, but I don't know if it's feasible this semester, which is that there's some interesting work going on in artificial life about measuring viability constraints, mostly in models using kind of like low dimensional dynamical systems. And I think that would be really interesting to try to get something like that for a CompuCell style simulation, because you could address problems like if you have an abnormal growth in a simulation, are there conditions where you can disproportionately decrease its viability by different measures without decreasing the viability of the healthy tissue? And that seems really valuable. But that also seems like a lot to bite off. So I need to understand more about the package first. The other topic I'm interested in is kind of the evolution of multicellularity and collective behavior in agents without nervous systems. So I was going through and I know there's some interesting work on slime molds. So that is another potential direction.
Those are both great topics. I mean, the problem you run into typically with evolutionary simulations with these agent-based models is that the individual simulations take a long time. and you have to run tens of thousands of generations to get your evolution to take off. And so, I mean, there's nothing, if you could do it in parallel, it's fine. So if you want to run well, actually at IU, maybe it's sometimes more complicated, but in general, running 10,000 simulations is not a problem. The problem is having to run 10,000 sequential simulations. You run 100 at a time, that's not so bad. But if you have to run 10,000 sequential steps to do your evolution, that's more of a problem. This issue of origin of multicellularity is something that's very interesting. And I think it's potentially something that could be modeled. As an example, the simple organism Volvox, which is related as a simple multicellular algae that's related to some unicellular ones. When there's a soma buffering hypothesis, where the idea is that the soma acts as a barrier between the germ cells and the environment. And because there's a paradox, of course, for people who haven't done origin of life, multicellularity is a paradox because only your germ cells replicate. And so that means all the other cells in your body die. And evolutionarily, at our stage, when cells break that contract, it's what we call cancer. But in early stages of life, when the difference between somatic cells and germ cells is not so great, you can imagine situations in which the somatic cells say, well, I'm going to become a germ cell again, and that wouldn't be lethal to the parent organism. And so there is a paradox about how you get from unicellular organisms where everybody reproduces to multicellular organisms where only a subset of cells reproduce. Slime molds are a good example of that. Myxobacteria are an example of this. And Volvox is another one. In the case of Volvox, it's been shown that if you put them into an environment that is highly variable or hostile, they get bigger. So the ratio of non-reproducing cells to reproducing cells goes up because the cost of carrying those non-reproducing cells becomes lower relative to the cost of dying for the whole organism. And so that's another hypothesis that one could potentially test in a simulation. I think it's a great problem. Okay, Wei Wei.
Yep. Hi everyone. My name is Wei Wei. I'm a research scholar from Purdue. So I'm working on the cell biology of plants. So we are working on the actin cytoskeleton of plants and its role in mostly defense signaling. And we are also working on the calcium signaling in response to a stimuli, either from a pathogen attack or a mechanical stimuli. And so we hope to do calcium modeling in plant cells and how the calcium signaling is spreaded from cell to cell. And we also might later modeling the actin responses like following the calcium responses. So so I'm mostly working on the biology side, and I almost don't know very little about the coding or the computational modeling. So that's the major things I hope to learn from this class, and I hope I can get help from everyone for the modeling. Thank you.
That's great. There's some nice papers. There is a paper in Nature published by Veronica Gruneson and Stan Murray, maybe 12, 13 years ago. And Veronica actually was briefly here as a grad student, but then she finished her PhD in the Netherlands, in Utrecht. And She uses the same kinds of modeling. She doesn't use our package, but she used the same kind of modeling methodologies that we use here. Now, she was interested primarily in root development, so in pattern formation and growth phenomena in plants. But it might be interesting to look at some of her models and ask the question, are there pieces of those models that could be used? Roland Merks, who's on the faculty in Leiden, professor in Leiden University, again in the Netherlands, he was my student. And he's developed modeling both using this kind of method and others of his own for modeling, again, more plant growth rather than damage response. But I think certainly modeling calcium signaling should be possible. And in the next couple of lectures, when we start talking about diffusion and reaction diffusion, I think you'll see some opportunities to be able to do that. So I hope, even if we don't get the model built that you want in the semester, I hope at least the methodologies and ideas will be interesting for you.
Yes, absolutely. Thank you so much.
Great. Andrew.
So I am a first-year PhD student in cognitive science, and can you hear me? Okay.
Can you hear me now?
Oh, yes.
Did that?
There's some
Very
static,
loud.
but you are audible now. Yeah.
Yes, we can hear you
Oh.
now.
Oh, wait, no, that's good. Whatever you just did is good. We could hear you say, oh.
It got better? Okay, great. So I am a first year PhD student in cognitive science and informatics. And I did my undergrad mostly doing from the data science perspective. And I was making a lot of descriptive models for, I guess, more biological projects. And I really kind of moved into having a stronger interest in the mechanistic models and like actually biological simulations. And I guess just brainstorming on like the types of projects that I'm interested in are like synaptic plasticity and how information kind of is spread across different, like whether it's neural networks or its information is exchanged from one cell to another and how that propagates. And I think one of what I actually recently got interested in is kind of this, I guess it's called a recursive functional programming perspective to, I guess, biology and genetics, where we have our programs where they have scripts, where they have scripts, but they don't modify themselves. But there are certain paradigms and certain, I guess, projects where you have programming scripts that end up modifying themselves in the same sense that genes and proteins interact with each other as data that is able to interact to modify other things and then be modified as well.
Well, we'll see how far we get during this semester. Those are topics that are pretty sophisticated. Python as a programming language, of course, is notorious because you could execute strings. So Python, I mean, Fortran doesn't allow you to do that in the old days. But Python, right, you could create a string and then execute it as code. So in principle, you could do that kind of recursive programming in Python relatively easily. I think as an organizing principle, one of the questions which is not the same but related in biology is how development works. Because again, in developed biology, there are no blueprints. And you don't have a concept of an overall plan. And you have to build organisms, or organisms have to build themselves from the inside out, as we talked about last week. And that means that the rules by which development happens are rather different from the rules with which we engineer systems. And so thinking about sets of rules that give rise to particular functional structures or behaviors is sometimes reasonably tricky. And it's not identical to the problems you've enunciated, but is parallel to it. And so I think that could be interesting. The other thing that could be interesting, potentially, if there's somebody who wants to work on slime molds, is that, and of course, when we say slime molds, we're actually not talking about slime molds, because ironically, dictyostelium discoideum are what are called cellular slime molds, which are not actually slime molds. There's a nomenclature problem there. But in dictyostelium, you have individual cells that have relatively limited capacity. They signal to each other when they aggregate. The aggregate of cells could do things that are not possible by the individuals. For example, the individual cells are not thermotactic. They don't move up temperature gradients. But the ensemble of cells are able to move towards light and towards higher temperatures. And the origin of that is pretty interesting. And again, it's a name I mentioned before, great modeler. Stan Murray, in his PhD thesis, did some very nice work trying to understand the origins of these collective, quote, intelligent behaviors from the signaling between dumb individual cells. And so that might be something that would be of interest to you, if it's worth looking at least as something that might be possible. Jackson?
Hey, I'm Jackson. I'm a junior at IU who's majoring in biology with a concentration in cellular biology. I came up with a couple ideas on what I might want to try to do a project for, but the one that I found the most information I thought was sickle cell vaso-occlusion, which is when the sickle celled red blood cells get clotted in the blood vessels. And it's mostly the interaction between the endothelium of the blood vessel and the sickle red cell. And I was going to maybe try to create a model of that.
Well, that's something we should talk about more. There's some beautiful, very sophisticated models that have been done in that general direction by George Karniadakis at Brown and then Roger Kam at MIT. They're pretty heavy. And there's also been some nice work on clotting, blood clotting more generally, by Mark Alber, who used to be at Notre Dame, is now in California. And he worked with some of the faculty up in the med school on modeling the formation of blood clots. And so I think as we go forward, we'll be able to see how far we could get with those ideas. But that's an interesting question. Blood flow is hard because the velocity of flow in blood is high, and that makes it computationally very expensive to model. But there are tricks for trying to do that, and we should see whether there's something in there that we can come up with that would be of interest to you. Again, when you hear people say that their main interests are biological, people who are more computational should take note of that, because those are resources that people can take advantage of as possible partnerships for projects. Next, I think we have Josh.
Hello, can everyone hear me? Great. So my name is Josh. I'm a master's student in the new Quantum Information Science Master's program at IU. My main interest is simulating quantum systems on quantum computers, which we can't really do right now due to hardware limitations and stuff. So I figured I'd start by learning how to simulate classical systems on classical machines. I did my undergrad here in math and physics, so I'm pretty bad on the biology side, but I've been programming for some time, and I took Vikram's class last semester simulating nanoscale systems, so I'm pretty familiar with a lot of packages we used. Probably won't use it in this class, but use a lot of lamps, running a lot of simulations on Big Red 3, so I would say I'm more computationally minded. And I registered in the class on Sunday, so I haven't really gotten a chance to take a look at a lot of projects, but the tumor vascularization thing looked kind of cool, and Jackson's sickle cell clotting also sounded interesting as well. So.
Okay, well, welcome. Again, I appreciate your patience with the issues with the startup of the semester.
No, that's fine.
And, I think it's great. As I say, this class often is very diverse, and that diversity, I think, can be a real strength. Jessica?
Hey guys, I'm from Purdue as well as Weiwei. We're actually on the same team. So I will also be studying the calcium modeling, hopefully. I'm a third year master's student, so Weiwei covered our project really, really well. So I don't really need to say much more.
And we should definitely, if we want to go forward with that, we should definitely draft TJ and Giuliano in to have those conversations. That would be great. And that last is Shelley.
Hello. So I'm also a Purdue student. I'm auditing this course just kind of to get an idea of what's going on. I'm definitely on the biology side of things. I'm working on the same project as Alexis, who isn't here today. But basically, the premise of what we're doing is that we're looking at like the coordination of an entire piece of tissue as a response to some external stimulus. So some background, I guess, is just that like when you wound like a piece of tissue, and this happens for every single organism, I guess, like basically down to invertebrates, there's this contraction, this swelling, contraction, relaxation response that I guess is present kind of everywhere. And when you interrupt that response, which can be done with like a cytoskeletal inhibitor, like a calcium inhibitor, that inhibits tissue healing later on down the line. So we're trying to figure out like what's going on there, and you know what the factors are, and kind of what's happening behind the scenes. And so Alexis is helping us kind of build a model, and I'm the one responsible for collecting a lot of the wet work data. I'm not that great at programming. Most of my experience has been limited to like Discord bots, the occasional Lua script. But I kind of wanted to get an idea of like what sorts of data are required for that sort of thing, what I guess I should tailor my experimentation towards, what kind of parameters I should be looking to collect. So yeah, happy to be here.
Okay, well, I appreciate that you're willing to give it a try. And I won't say that there's no programming in this course, but one of the reasons we built the software we did was to try to minimize the amount of programming that's necessary. And you can tell us as we go forward how successful or not we are with that.
Yeah, I mean, like so far, just kind of looking at the demonstrations, it seems pretty straightforward. So I'm sure that I'll at least be able to wrap my head around the general principles of how it works, if nothing else.
Right. Okay, I think that's everyone then. Thank you for taking the time to introduce yourselves, and I'll look forward to meeting with you and discussing as project ideas happen. And I encourage people again to begin to reach out. Maybe in the end you wind up pairing up with somebody, and you wind up neither doing the project you dreamed of, but both of you doing a project that's interesting. And a semester isn't so long, so the level of complexity of what you could do in a semester is limited, but the goal at least is that you do something which could be the basis of something in the future. Whether you go forward and do it or not is another question, but at least you something that is a stepping stone towards the ability to do things that you're interested in. So coming back to the slide deck, does anybody want to make a comment about the homework problem 2 or 3? You don't have to, but I wanted to give people an opportunity. If somebody had a result for either of those homework problems, I thought I'd give people a chance to show them off if they liked.
Something that I wanted to ask was, so for problem number 3, I was having an issue in nanoHUB where I think I was able to answer the questions, but most of the time I couldn't get the simulation to run past about 250 to 300 Monte Carlo steps. So if anyone was able to get it to run further than that and would like to share kind of like what that looked like, I would be interested in seeing it.
So that's one. Giuliano, do you know what happened to that simulation? Because it ran fine. The last commit of that simulation was July of 2021, at which point it was working fine. And there have been some issues with it. I think probably the thing that we do there is reference you to the paper, which has examples of it.
Yeah, I did end up looking through the paper.
And we'll do our best to get that debugged, because it's a bit mysterious to me what happened. Normally when code works, if it stops working, it stops working because somebody did an update of the system software. My guess is that what happened was somehow a library that code depends on in nanoHUB was changed. And so I appreciate people reporting that. I tried running it myself. In my case, it didn't crash, but some of the graphics didn't work properly. And there's actually today, the example I'm going to have you do is going to have some of that as well, where you'll see some graphics issues in the nanoHUB deployment. I don't understand where that problem is coming from. But it's very helpful to us, because if it's not reported to us, we don't know it. So the best I can do is to say that we'll work on it. Something that I will say also with homework problems, and it could come up this week already, is that I don't always know exactly how far we'll get during a lecture. And so sometimes then I write the homeworks up beforehand. And so if there's a homework problem that covers material that we didn't get to in class, you're welcome to try it. You've got the slide deck, so you could look at what the lecture would have been, or you could try it based on online materials. And that's not a bad way to prepare for the next class. But it's also possible to kick it to the next week. If you say, I'm not ready to do this problem, that's fine to respond that way. In this case, I think we'll maybe save digging into that viral replication model until Giuliano and I can look at the code online and see what's going on with it, because Joel did bring it up the other day, and we ran it, and we were seeing these problems in terms of the graphics and in terms of it hanging, and we weren't able to debug it so far. I'm not sure what's the problem is. It definitely would run correctly on your desktop, and so if you have a desktop deployment, it'll work. I realize that defeats the purpose a little bit of having the online deployment, and so I wouldn't require people to do that. And if people had, if that happens, and your answer in your homework problem is, I ran it and the code crashed, I'm not going to take off points because our code didn't work. So I understand it's frustrating for you, but I don't want people to spend tens of hours trying to get code running that doesn't run. And so please understand these homework problems tend to be quite open-ended, and there's nothing wrong with spending more time on them if you're interested. But the expectation of the class is not that you're going to spend 30 hours on a homework assignment. The project assignment is heavy. There's no question about that. But the homework assignments are not meant to be that heavy. And so if a problem seems very, very hard, please do approach me and ask, is it supposed to be this hard? Maybe I made a mistake. Maybe there's a typo in the problem, or maybe there's an error in the software. But you certainly, as far as, I don't think anybody's in this class for a grade. You don't take this class because you want a grade. People come to this class to learn. But I also don't want you wasting your time. And so the moment working on a problem stops being a learning experience and starts becoming frustrating or a waste of your time, at the very least, please let me know, and we'll do our best to fix it, at least for next year, so that I don't reassign a bad problem. So I do appreciate that. The key ideas here were really, it was meant to be, in a sense, a very trivial problem where you played with a simulation online that worked and had some results where you could see the difference between a situation in which you had propagation of disease from a situation in which the immune system controlled the infection. And that was really the goal of the exercise. And that Rmax multiplier was simulating treatment with remdesivir, which blocks viral replication. And so that exercise was really to explore a little bit about immune response and antivirals. And I'm very sorry that turned into a problem for people. It wasn't intended that way. And did people have problems also with the cell growth and division simulation was the one we did in class, so that should have worked for people. Did people have problems with that, or did that work all right? That worked okay. All right. So again, one of the things that we're going to face in this kind of class is that it's not like an introductory engineering or physics class where there's a big textbook that's had hundreds of thousands of hours of people working on it. And so mistakes happen. And the best I could do is to try to address them when they happen. And we will try to figure out what's wrong with that simulation, because it's a simulation a lot of people like to run. And so having it break was a bit of a shock. So I apologize for that. Okay, so the first thing I want to do, if everybody can get, if you're running desktop, you can tune out perhaps for a few minutes. But if you are on nanoHUB, I would ask people to launch their nanoHUB deployment. And I want to go over one of the annoyances of nanoHUB that we're going to face, which is that uploading and downloading files has to be done from outside of CompuCell. And this is a security restriction within nanoHUB. And so I want you to show you how to upload and download files. It shouldn't take very long to do that, I hope. Giuliano is here in case there's a problem, which is good. And so I would ask people to launch their nanoHUB deployment and then follow along. And I also have put into the chat, it was the last chat message that I had, a link to the file we're going to use as a demonstration to upload and download. Okay. So you'll need to pull that file from the Google Drive to your local machine so that you can then upload it. So we're going to use a simulation that was actually written by a grad student here, one of Paul Macklin's PhD students when he took this course a few years ago. It's a simulation of the colonic crypt, of stem cell development in the colonic crypt. And the first thing that you'll need to do is to go to that link that was in the chat for the Google Drive and grab the zipped folder.
Could you resend the link in the chat? It's not coming up for me. I think maybe because I joined after it got sent.
Okay, here we go. Does that work for people? If people are having trouble with that link, let me know. I don't want to spend too much time on this today. So if we wind up getting stuck, I may kick it into people trying it at home. But I do want to make sure that at least it's possible, because we're going to need during the week ahead to be able to upload files. And so downloading them as well, because we want to be able to do backups locally on your local machine. Were you able to, did that link work for people? Okay. So don't unzip it. Just pull the file down and give it and put it somewhere where you remember where it is. So just check to make sure. I'll use that poll as usual, just to make sure that everybody is able to do it. Does anybody need more time with that? Looks like most people have it down. Okay. And so the next thing that you have to do is you're going to have to use the Jupyter function, the Jupyter Notebook function in nanoHUB. And so I have Jupyter Notebooks running already because I'd already launched it. But if you don't have it, it's one of the things you should bookmark in nanoHUB. Please log into nanoHUB. Go to your dashboard. Click All Tools in the dashboard. I'll do this with you. So click All Tools here. Search Tools. Jupyter. See if it works. And you'll see Jupyter. We don't want the Jupyter GPU version. We just want Jupyter Notebook here. And you probably want to bookmark it by clicking the little heart next to it so it's easy to find. Does everybody have that work running? Were you able to get to that? Okay, great. And so now you want to launch it. So I have it running already, but if you're going to launch it, you just click the launch icon here. I'm going to go back to the session I have. I hope. Open. See if it'll do that for me. Yes. And you should get a window that looks like this. And the key thing here is that there is an upload button. So click on upload. You'll have to navigate to find the zipped file that you want. Here it is. It'll obviously be in a different place for you. And then you'll have to click upload again to actually confirm the upload. So did that work for people? If people are having problems with it, by all means speak up. I don't want to find out, gee, nanoHUB disabled the upload feature or something like that. It's not an impossibility. So we want to make sure that we know that it works, because if it doesn't, we have workarounds for these things, but they're not as convenient. So this is the way we want to go. All right. So did that, if everybody's got that, then you're okay. So now, once you've got the file uploaded, we can go back to our CompuCell release. Go back to CompuCell. Boy, that looks pretty, doesn't it? Let me try that again. So you want to launch your CompuCell. And now, that's actually a question, Giuliano. Do we have to go to Twit for to unzip on nanoHUB, or will it open will player open the zipped file now? You're muted. No, your mic isn't working. So inside of CompuCell, we want to go to Twit, the editor, the model editor. And then inside of the model editor, we can hit Open Project. And you should be able to find the file that you just uploaded. Were people able to find it or not? Everybody got it? Mine had already been unzipped, so, okay. When you click open, it may throw you an error, a warning message saying, we'll unzip into such and such. And that should be clicked OK. It may give you a second warning message. And once it's unzipped, you can open it. And you should be able to also then run it. So when you've got it, you should be able to open it, find the model, click on the CC3D. It should look like this. There's the simulation. And now I can open in Player with a right click here. Open in Player. And now I can switch back on the bottom here. I should have a tab for the window with Player. And it should launch the simulation. Did that work for people or not? Yes? Joel, did that work, or are you running a desktop?
No, I'm running on nanoHUB. It worked. It's taking a while for the simulation to run though. Like everything is static right now. It's the same as yours. It's fine.
No guarantees on how long the simulation runs. The goal here was simply to get the thing uploaded. So this is not the most convenient thing in the world, but it takes about a minute, maybe 2 minutes to do it. And so once you've done it a few times, it's not too bad. I hope. Remember that if you want to switch between Twit and Player, you've got the tabs at the bottom here to switch back and forth between the two functions. And the simulation should look something like this. So that was the point of the exercise. I'm not going to, in principle, there are a lot of things we could do to explore the simulation, but for today, we're just going to run it that way. The other thing that we might want to be able to do is to be able to download a file. And downloading a file, we're going to do the same thing that we did here, which is we can go to the simulation and twit it. We can right-click here on the simulation name, click zip it, and it will give me the option of saving this file somewhere. And let me give it a unique name just so that I don't overwrite something. So you can save the file. And then if I switch back to Jupyter Notebooks in my dashboard, assuming it's going to let me do that. I can now select colonic crypt here. I need the zipped one. Let's see where the zipped one went. Here. I'll pick one of the zipped files. Doesn't matter. Here it is. This is the new one. Here. Example colonic crypt. I click on that. Go up to the top. Your workspace will be a lot more empty than mine. Click download, and it'll download it to my downloads folder. So why don't people just make sure that they can download things? In principle, when you do simulations and save them in your workspace, they should be persistent from week to week. In practice, it's always a good idea if you're editing things and have code that you like up on nanoHUB, you should always, before you end your session, do a download of it. Because it's a public server and there's no guarantees on that. So, is anybody having trouble with the download, or did that work for people? Okay, to review again. Inside of twit, you select the topmost name, in this case, colonicrypt.cc3d. You right-click on it, say zip it, give it a name, and save it to your local workspace. Then you have to jump out of CompuCell into Jupyter, find the file that you just zipped. Here's some examples. Cancer evolution is an example. Cascade chemical reaction and so on. Select that. Go back to the top of the window, and then you should be able to select a download inside of Jupyter. And it looks like with the Jupyter Notebooks implementation they have, that you can only download one file at a time. I notice if I select the second box, the download button disappears. So you can only download one file at a time. Okay. Does everybody have that more or less satisfactorily? We're not going to really need it today, but I want to make sure that that's something that it's an annoyance, of which there will be some, but it's at least people have that basic ability. Again, this is specific to nanoHUB. If you're running locally, you won't have this issue. Okay. Anybody need help before we move on? Okay. So now I want to talk a little bit about CompuCell. And I'm going to be covering a couple of things. One, how we represent cells. We talked about this very briefly last week. How we represent cells and how they move in the lattice, and how we represent what they do. And there's going to be a little bit of mathematics and a little bit of physics today. And if the mathematics and physics are not meaningful to you, it's okay. You're not going to need them, at least in any detail in this class. But for the people who have a little bit more math or physics background, I'm going to talk about it a bit. And feel free to ask any questions that you feel would be helpful as we go forward. And so again, we're going to talk first about how cells are represented, and then how we describe what they're doing, and then how they evolve in time. So the first thing we have to think about is how CompuCell represents space. And the way CompuCell represents space is on a lattice. So essentially, it's like a video camera pointed at something. Each pixel in the video camera corresponds to one region of space that's being modeled. If we think about the universe more generally, you can imagine in a two-dimensional universe, you have an x-coordinate and a y-coordinate, and there's some extent, the size of the region that you're going to be modeling. There are modeling packages where you don't have a fixed volume of space that you're going to model. Although typically any package that does diffusion fields has to have a space volume associated with it. And CompuCell allows you actually to move your space. You can add empty space to your simulation or take it away if you need to. Now, that's not something we do very often, but it's possible and it works in CompuCell. But typically, we're going to represent space using a Cartesian coordinate system, x, y, and z coordinates. We're going to be on a lattice, and so our typical spatial volume will be a rectangular parallelepiped, either a rectangle or something that looks like a brick, and we'll have to specify how much space we have in the x direction, the y direction, and the z direction. CompuCell does not do a great job of, you can actually specify units in CompuCell, but people typically don't do it. And so we typically do unit conversions by hand. So we'll write down somewhere that one voxel or one pixel is 2 microns. And then we have to keep track of that ourselves. And that's something that we have to think about. In general, the simulations we're going to be running will use the one voxel will represent 1 micron to about 5 microns of real space. But there's no reason you couldn't have a voxel represent a centimeter or a voxel represent 1/10 of a micron if you wanted to. Something that may be a little bit less familiar to people. First, the the space can be two-dimensional or three-dimensional. It just depends on what you set your z coordinate to be, your z depth to be. A z depth of 1 is two-dimensional. A z depth of more than 1 is 3. There is something called a 2.5-dimensional simulation where you have sheets of 2 dimensionals which are stacked on top of each other. That's actually going to be implemented in the newest version of CompuCell, but we don't have it out yet. Something that will be maybe a little bit less familiar to people, although if you've done physics or programming on lattices, these may be more familiar, is that besides the normal Cartesian grid, square grid that you have, you can define a hexagonal lattice. And hexagonal lattices are nice because you don't have the symmetries of a square lattice. So they behave a little bit better computationally. The other thing that we're going to have, which you're going to run into repeatedly and may take some getting used to, is that there's going to be a concept of an interaction range. And so on a square lattice, things like distance are a little bit hard to measure on a square lattice. If I was in a real Cartesian world, the distance between point xy, x1, y1, and x2, y2 would be x1 minus x2 quantity squared plus y1 minus y2 quantity squared, all square root. Just the Pythagorean formula. On a lattice, distances follow the lattice, and so the scaling isn't so simple. And so you will find that there can be small annoyances. Anybody who's done imaging and had to do dithering is familiar with this kind of problem. This is a intrinsic problem of lattice models. One of the ways of dealing with that is to say that this black dot has nearest neighbors labeled 1, north, south, east, and west. Its second neighbors are northeast, southeast, northwest, and southwest. And so you have 4 nearest neighbors in 2 dimensions, 8 second neighbors in 2 dimensions, and you can go to third neighbors and fourth neighbors. As you increase the interaction range, you're basically fuzzing out the granularity of the lattice. The simulations will take longer to run, and you may get funny non-local effects, but they're also going to behave better mechanically. And so we're going to do occasionally, you'll be asked to set the interaction range to 2, to 3, to 4. And sometimes you'll be asked to play with that and see what happens. That's clearly not a property of the universe or a property of real experiments. It's a property of this particular simulation methodology. And it's something that typically we will try to minimize the amount of time you have to play with it, but you may run into it. On a hex lattice, you have 6 nearest neighbors. And then as you go out, you have more. When you go to 3D, the number of neighbors is different. Doing 3D on a hex lattice, you have to do some mental gymnastics to keep track of the adjacencies. Fortunately, CompuCell lets you do that automatically. CompuCell is set up so that if you want to do something crazy like say specify 12th neighbor interactions, you can do that. Joel, question?
Yeah, just by looking at it, the standard is 1 neighbor for the calculation, right? Is it set to 1 neighbor?
We typically run at 2 or 3.
Oh, okay. Because I was about to ask if the hexagonal one is not more computational expensive since you have 6 neighbors on one, and on the other one you just have 2, or actually 4.
Right. So the computational cost difference between having 4 neighbors and 6 neighbors is not large. The computational cost between having 4 neighbors and 96 neighbors in the 4th neighbor 3 dimensions is fairly large. In general, you do better on a hexagonal lattice computations are smoother, and you can go, you might need a fourth neighbor range on a square lattice to have the same convergence of behavior that you get with a second neighbor on a hexagonal lattice. We will tend to not use hexagonal lattices because thinking about them is harder, because you have to think about adjacencies are a little more complicated. But when you're doing serious simulations, it's often worth thinking about that. I will try to minimize the amount of time we spend dealing with the minutiae of CompuCell structure. And this is something we're going to run into fairly soon. For people who don't want to think about it, you can ignore it and simply know that we'll be telling you periodically what interaction range to use. But for people who've had, especially if you've had physics of statistical physics, this kind of interaction range analysis is something you probably might run into if you do the Ising model, for example.
Thank you.
Okay. So when we run a new simulation, remember last week we used Wizard to create a new simulation. When we do that, Wizard will ask us how big the lattice should be in X, Y, and Z directions. So 256 by 256 by 1 is a two-dimensional simulation, 256 by 256. But I could make it 256 by 256 by 10, which would be sort of a pancake. I can adjust the boundary conditions in each direction to be no flux, which means that the world ends at the boundary, or periodic, in which case you reconnect in a toroidal way. So something that leaves on one end, if a cell crawls off of the lattice at one end, it comes back on the other end. Because the boundaries aren't real, usually, we often use periodic boundary conditions. Of course, the real world isn't periodic in that way, so we have to be aware of the consequences. If we look in the CC3D-ML, and probably it would be better for me to go back to, let's see if I can go back to my CompuCell example here. So that we can see this. If we look at the bad choice. If we look at the font sizes, if we look at the XML simulation, we will see a section in the XML called POTS, and it's cut off here because of the window size, which will say basic properties of CP-MGGH algorithm. And there'll be a line that says dimensions, X equals 100, Y equals 100, Z equals 100, or X equals 256, Y equals 256, Z equals 1. So that specifies the size of the lattice. And then the neighbor order, which is precisely what we were talking about, which is this issue of adjacency. And why we need that, I'll come up, we'll talk about in a few minutes. And then the options of having each direction be periodic or not. So typically you don't have to edit that by hand, but if you need to, the tags are fairly self-descriptive. Okay. So that's how we create a space in which we're going to do our simulation. And now we have to create cells and say specify what they're going to do. Now, here's a picture of some cells. These are stained. This is probably fixed tissue, probably an epithelial tissue. They've used a stain for, it looks like this may be an actin stain in red, would be my guess. And in green is probably histone stain for the cell nucleus. And so the cell membrane is outlined in red, and the cell nucleus is outlined in green. And how would we represent that computationally? Well, in what are called cellular automaton models, there's a lattice, and each pixel in the lattice, or voxel in the lattice, corresponds to one cell. In a center model of the kind that we use in our Mechanica software, or Paul Macklin uses in his PhysiCell software, each cell is represented by a point and an interaction range around that point. So the cells don't have any intrinsic shape. There also are what are called vertex models, where we represent the boundaries of the cells, the membrane of the cells. Each vertex between cells is represented as a point. Each edge connecting two cells is represented as a link or a spring. And so that would be a vertex model representation of the cells. And so this is all starting from the same image of cells, and we've come up so far with 3 different computational ways of representing it. In our case, what we're going to do is, again, think about taking a photograph of this with a digital camera. And so we're going to imagine that these cells are laid out on our lattice, and the lattice constant is going to be set to be smaller than the size of the cell. If the cells are typically 10 microns, then our lattice constant might be 1 or 2 microns, so that we have 5 or 10 pixels across. And in this case, this one cell here will be called, all the pixels that overlay that cell in the image are then given a separate identity. And that's how we're going to work. In a sense, it's the most naive way you could think about this kind of thing. One of the things that we're going to find, then, is that the volume of a cell exists, it's an object in a computational sense in the simulation, but the boundary of the cell is just a region of the lattice where one lattice site belongs to one cell and the adjacent site belongs to another. It's not an object. And that can actually cause a little bit of an annoyance later on, because it means there isn't an object, computational object, to attach things to when we're doing computer simulations. And we're going to do a lot of work to deal with that problem in the future. One of the results of this is that the volume of a cell is well defined. It can count how many voxels belong to the cell. But the surface area of the cell is going to depend on how I measure it. If I have nearest neighbors, I can count. The surface area is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, and so on. If I had second neighbor interactions, this one pixel would have 1, 2, 3, 4, 5, 6, and so on elements of surface. And so surface area is going to depend on the lattice description that we use, whether we're hexagonal or square, and what the neighbor ranges are. Within a simulation, if you don't change the lattice, you don't have to worry about it. But if you decide, gee, I've been using a neighbor range of 2, and now I want to use a neighbor range of 3, then some of the numbers that you measure of surface area will change. That's something we have to think about. Each cell in a simulation has a unique identity. We call it a cell index, and it's just the number of the cell. And that number isn't meaningful. We'll have cell number 1, cell number 2, cell number 3. If you're an experimentalist and you use image J and do image segmentation, you'll say this is cell number 1 in the image, this is cell number 2, this is cell number 3. The thing that's important is that every cell has a different number. And in the equations that I'm going to show you, and again, I'll try to not have too many equations in this course. Today, there'll be more than usual. I promise we normally won't have that many. We use an index, and it tends to be labeled sigma. And some of these features of variable names and things like this, some of the terminology is borrowed from physics, which isn't necessarily so meaningful in a biological context. But in any case, by default, when you give the cell index, you use the Greek symbol Greek sigma, letter sigma, lowercase sigma for the index. Each one of these regions could also represent something other than a cell, really represents a volume of material. We could have a region that represented the nucleus of a cell. We could have a region that represented the cytoplasm of a cell. We could have a region that represented a volume of blood. And so really, these are what are called generalized cells. These are simply regions of space which have a unique index that we can identify and give properties to. As I mentioned, automatically, when you have a cell, you have a volume associated with the cell, because the number of voxels that have that index is well defined, and that's the cell volume. The surface, as I mentioned, is dependent on the lattice we use, but typically there's a concept of surface which we'll call S. Now, there isn't a consistent way to talk about volume and surface when we want to do 2D and 3D. And so we're going to have to get used to the fact that in 2 dimensions, what looks like the area of the cell we call the volume, and what looks like the perimeter of the cell we call the surface. And that's terminologically what we're stuck with. We're also going to have something which comes from biology, which is a concept of a cell type. And that's usually denoted by the Greek letter tau. And that's going to be useful because we want to say that cells number 10, 15, and 20 are muscle cells, and cells number 11 and 77 are blood cells. And so we want to be able to have a concept of a type of cell. When we want to specify what the cells are going to do, we're going to have to identify what properties the cells have, what the state of the cells is. And we're also going to have to specify some set of rules that describe how those cells change in time. And this is going to get rather physicsy. And again, for people who don't find this comfortable, we'll come back to it, and we can also, you don't need to have a lot of detail on it. Basically, a lot of the things we're going to be doing are going to be trying to map concepts in continuous space onto the lattice that the cells live in. So cells in CompuCell are computationally objects, and they're going to have a set of attributes. Those could be things like volume, surface area, whether they're touching other cells, whether they have a concept of directed movement, and so on. Those are going to be implemented. In the real world, things move in response to forces. Things don't move if there's no force acting on them. Things move if you apply a force. when you come to the mathematical and computational realization of movement in simulations, in models and simulations, you have to make a choice about whether you represent the forces acting on objects directly, or whether you use a physicist's approach, which is to represent forces in terms of potential energies. And one of the things that some people find CompuCell a little bit confusing about to begin with is that CompuCell most of the time uses potential energies instead of forces to represent how things are going to change. And the reason for this is that a force is a vector quantity and a potential energy is a scalar quantity. And so it's simpler mathematically and computationally to represent things in terms of potentials. And so we're going to see a lot of times we're going to see properties or behaviors of the cells represented as what we call effective energies. And the model specification will essentially consist of writing down a long list of effective energies that implement the behaviors that you want. As I said last week, these kinds of simulations, nothing exists unless we assert it. And so cells will not have a volume, a surface, length, a contact, a behavior unless we specifically specify it. And so we'll be seeing today, I don't know how far we'll get during this lecture. I don't think we're going to get through all of the material that I had originally planned, which is fine. I'd rather slow down and make sure that people are able to follow. We're going to go into each of these concepts one at a time and play with it and see how we implement these various aspects of cells. And it may not seem very exciting to say that cells have volume, but again, computationally, we have to decide how we're going to implement that. And so until we give cells a volume, they don't have it. Within CompuCell, the code that implements a particular effective energy is called the plugin. And so if one wanted to build extensions to CompuCell, you typically are writing things called plugins. Okay. Another thing that we're going to have to get used to, and again, it may be a little bit less obvious if you haven't worked on these things in a physics context, is that some of the things that we think of as intrinsic properties of an object are actually going to be implemented as behaviors. And we're going to talk a lot about some concept called constraints. And a classic example of this would be the volume of a cell. If we had a box of 100 microns by 100 microns by 100 microns, and we asserted that we had 1 million cells in the box, and we asserted that each cell had a volume which was 30 microns by 30 microns by 30 microns, we'd be overfilling our box. And so we're not allowed to specify in the model what the volume of a cell is. We can specify what the target volume is, what the equilibrium volume of the cell is. But the actual current volume of a cell is going to depend on the environment that it finds itself in. And this is something, again, that may take some getting used to. Once you've worked with it, it'll feel, I hope, pretty natural. But think about it this way. If I give you a bunch of balloons, and you have the balloon out on your desk, the balloon will have a volume. If I now tell you to take 100 balloons and pack them into an Amazon carton, you'll have to compress the balloons in order to get them to fit. And so the volume that they would have on the desk is not the volume they have when they're packed into the box when they're compressed. So the target volume of the balloon would be the volume the balloon has sitting on your desk, but its actual volume will depend on how much you're pushing on it. Or in principle, you could be expanding the balloon and making it bigger as well. And so that's a first way of thinking about this idea of what we'll call a constraint, an elastic constraint. And I guess I want to skip over this, except to say something here, which is that we're going to see again and again in CompuCell and in the way we think about models, something called a target X, a target volume, a target surface, a target length. And we're going to see, paired with that, whatever the actual current volume, surface, or length of the thing is. The target will be what that thing would be if there was nothing else happening in the world. The actual result is what happens given all of the interactions and constraints on the system. And this would be true in biology as well. A cell might by itself go through a cell cycle and divide, but be in a situation where it can't divide. It's under pressure, it has contact inhibition, something else is preventing it from dividing. And so this idea that there is some intrinsic thing that the cell is trying to do that may be changed by its environment is something that's very biological. Some of the physical constraints that we're going to run into in CompuCell in this kind of modeling may not seem so biological, but the basic idea that the potential of the cell is not always realized or is changed by its environment is something that's a pretty biological concept. When we're talking about constraints, if it happens that the target and the current actual equal each other, the target volume is equal to the actual volume, then we say that the constraint is satisfied. If we're used to numerical optimization, constraints are called penalty functions. And if we're a neural network, artificial intelligence, machine learning person, those constraints are called loss functions. So the term constraint has got different names depending on the world you live in, but it's a concept that you run into in all of these different book fields. So what is a constraint? A constraint is a function of the object's property and its target property, and it has the property that property is a bad choice. It also has the characteristic that when the property and the target values agree with each other, its value is 0, and that when the actual value moves away from the target, its value goes up. So conceptually, if I think about the x-axis here being the actual value property, the y-axis here is the value of the constraint. If my target agrees with my actual value, the function is 0. If my target and my actual value are distinct, the bigger that difference, the bigger the value of my function. A classic example of this is suppose that I have a hemispherical bowl and I take a marble and I drop the marble. Gravity will pull the marble down, and eventually, when dissipation happens, the marble will land and stop at the bottom. So you can think about a constraint as a shape in some abstract space that's pushing you in the direction of satisfying the constraint. Another way to think about this is that the constraint behaves like a spring, pulling you towards the actual value being the same as the target. And we will typically use a potential energy function that is quadratic in the divergence between the actual and target value. There'll be a spring constant, K, which we'll normally use the Greek letter lambda to represent. And this quadratic constraint, also called a harmonic constraint in physics, is going to have the property that it's always going to pull things back to their targets. And it's what's called a soft constraint because it doesn't pull very hard. And so we can imagine here's our object. It is at equilibrium, or this constraint is satisfied when it's in this along this middle line. I pull it away from the middle line. It creates a force that pulls it back. In this case, it overshoots and oscillates until it finds its equilibrium. This potential energy, H spring, equals Kx minus lambda L squared, quantity squared, creates a force minus Kx minus L. And the key thing is that the restoring force works if the spring is too long, it shortens it. If the spring is too short, it makes it longer. And so this methodology creates forces that act to try to satisfy the constraints. If you write Newton's law equations with damping, you'll get something similar from that. You'll get constraint where you get satisfaction of the constraint. Dissipation is important here. If there were no loss of energy, my spring would keep oscillating around its target. It would never reach it. We need dissipation. And so it's important from a physical point of view that these are dissipative systems. These constraints behave like physics potential energies, and they're typically called effective energies. The reason they're called effective energies is that they may or may not correspond to actual physical energies in the system. They have the mathematical structure of energies. And so we're going to see again and again, when we want the cell to have a given volume, we write a constraint on its volume. When we want the cell to have a given surface area, we write a constraint on its surface area. When we want the cell to have a given shape, we write a constraint on its shape. This lambda, the strength of that spring constant, tells us how strongly we are going to demand that that constraint is satisfied. Alternatively, we're going to say how expensive is it if we fail to satisfy the constraint. And people who've done machine learning will be very familiar with the idea of having weights for components of the loss function. The force for any constraint with this quadratic property is -2 times lambda, the strength of the constraint, times the distance between the target and the current value of that property. And what's wonderful about this is the further away we are from being satisfied, the harder the system will try to push us back. So it's always going to try to push us back to satisfy the constraint. And the other thing that's wonderful about this is that we can write any property of a system in the form of a constraint. If I say the cell is supposed to move 3 feet north in 10 seconds, and then is supposed to become bigger, and then is supposed to change color, I can write a series of constraints that will enforce all of those behaviors. They may be mathematically awkward and ugly to write down, but I don't only have the ability to specify a spring. And this constraint formalism allows me to create effectively virtual springs that pull my simulation to do anything I ask it to do. And that's why this is such a powerful way of representing physical reality. Again, Joel, question.
Yeah, if I may, I think I ran over that on my homework because I was playing with the values, and one of the values that I play with was with the lambda volume. And my interpretation at that time, I even looked at the manual, saw that function that you just showed, and my interpretation was because it was affecting the target volume somehow, I doubled it to try it out. And so I noticed that the cell was growing twice as fast, right? But now with your explanation, I don't know how to like correlate the two because I had a simpler explanation in my head.
So there are two components, and this is, depending on how far we get in class, we will explore exactly that question. When I have a constraint for the volume of the cell, I have to specify a target volume, and I have to specify the strength of the constraint. If you're a physicist, the strength of the constraint is the inverse compressibility of the material. In the elastic constraint that we're using for volumes is equivalent to the ideal gas law. The cells really do behave like balloons with gas in them. And the compressibility of the gas, the bigger the compressibility, the easier it is to compress. The bigger the lambda, the harder it is to compress. And so lambda is 1 over the compressibility. It's, in fact, the Young's modulus, if you're used to materials, of the material. And so what this is telling us is not the speed at which your cells grow in the simulation that you did in the homework was determined by the rate at which you changed the target volume. What lambda volume tells you is that if you're at the strength, if you've satisfied the constraint and you move away from it, how fast do you come back to it? And so it's basically telling you how big the restoring force is when you move away from the target. And what's going to happen when we write simulations is we're going to have to tune things so that all those restoring forces are about the same order of magnitude. Because if I have one force that's very large and a bunch of other forces, the ones that are small aren't going to matter. And I'm going to typically want all of those things to be acting simultaneously. And so that's where the tuning of these models is going to come in.
Thank you.
Again, I realize that for people who haven't done the physics of this, and even if you've done the physics of materials, it's not always taught in this way. And so if this seems alien, I'll ask you to be patient, because I think once you start working with it in computation, you'll see how it works pretty quickly. And so even if the mathematics are unfamiliar, once you start playing with it experimentally, it should become more reasonable. Okay. So again, the key thing about this is that we can build a simulation by specifying a series of constraints that add together, and then provided that we weight those constraints appropriately. And again, people who do machine learning will be used to the fact that you have to adjust the weights of your loss functions pretty carefully. Provided that you adjust your weights of your constraints appropriately, you can build a simulation that will implement all of these constraints simultaneously. And so we're going to see models specified as sums of these constraint energies. The next thing that we have to think about is what are our basic constraints. The two we're going to run into today, I hope, and I'm going to take a break pretty soon to let people catch their breath, are volume and surface area. Most cells are going to have a volume. You can imagine the material amount of protein in the cell, for example. And so we're going to write H volume, effective energy from the volume, is going to be a sum over all of our cells of the strength of the constraint, lambda volume. That could be different for every cell, although typically we won't do that. Of the times the current volume of the cell minus the target volume of the cell, quantity squared. This gives rise to a concept of a pressure. Again, think of the cell as a balloon. The pressure is that lambda times the difference between the current volume and the target volume. If you want to think about this biologically, you could think about the osmotic pressure of a cell. What you'll get, though, is that in reality, the osmotic pressure of the cell is much higher than we're going to implement using our lambda. We're going to make our cells more compressible than they would be in real life. But cells are compressible. If you squeeze on a cell hard enough, it'll shrink. And if you put it in a situation where water gets pumped in, it'll swell. So it's not actually as unreasonable as it might seem. We're often going to define also the surface of the cell, lambda surface. Think about the amount of membrane the cell has. Current surface minus target surface quantity squared. I won't go into the details of the surface volume ratio. Now, will be a homework problem where you can explore that a little bit without worrying too much. But if you think about cells, you can have cells that are very spherical, where the volume is large compared to the surface area, or you could have a cell which has very floppy surface, where the surface area is large compared to the volume. The way you could think about that is thinking about inflating a beach ball. I don't know, or a balloon is less good because balloons get rigid pretty fast. But if you think about a beach ball, if I start out with a beach ball and I blow a little bit of air into it, it basically stays flaccid, and you have a lot of surface of a small volume. As I blow it up, it rounds up. If I keep blowing air into it, the surface is not very stretchable, and so I'll wind up with something that's quite rigid and incompressible. Okay. One thing, as I mentioned, that we'll have to deal with the surface area is that you're on a lattice, and so the counts of surface area are not quite the counts you would get in the real world because you have stair stepping because of the lattice. So we've defined forces. Now we have to define how things move in response to forces. In the real world, we're taught in physics classes that objects obey Newton's law. F equals ma. Force equals mass times acceleration. As anybody who ever read Aristotle would know, Newton's law implies inertia, that once something starts moving, it keeps moving. Whereas in reality, we all know that if you stop pushing on something, it stops moving. Now, the idea of friction was developed in the 19th century to explain this, but for thousands of years, people assumed that in reality, the law wasn't that F equals ma, that force is mass times acceleration, but that force is equal to mass times velocity. If you push on something, it moves. If you stop pushing on it, it stops moving. At the scale of biological cells, inertia doesn't matter, except in very special cases. In particular, in blood flow, you have inertia. But in the movement of cells and tissues, there's no inertia. If you stop pushing on something, it stops moving. And so what we need in our world is going to be a dynamics in which things move at a velocity that's proportional to the force that's applied to them, and not that accelerate proportionally to the force that's applied to them. And that's called perfectly damped or overdamped movement. It also creates the idea of a drag, which replaces the concept of mass in Newton's law. And the drag function is something that mathematically is a little problematic in CompuCell, but it's there. Okay. In reality, cells move actively because of cytoskeletal rearrangements, primarily due to myosin and actin. Actin can provide extensional forces, although those are pretty weak. Myosin primarily provides contractile forces, and the combination of contraction and expansion gives rise to cell movement. If we look at the membranes of cells, the membranes of cells typically are fluctuating. Sometimes they do things that are rather complicated. They'll send out fibrous protrusions and pull on each other, filopodia. But our basic idea here will be what's called amoeboid movement, where the cell membrane fluctuates randomly on some scale, and the cell's membrane explores the volume around it. And the way cells move in CompuCell is based on the idea of amoeboid movement. How does a cell move? Cells move in CompuCell by taking pixels or voxels in the cell lattice that belong to one cell and reassigning them to another cell. That's called a voxel or pixel copy. And we'll come back to that. And the dynamics of the simulation are given by changing the probability of pixel copies happening based on the energies, the effective energies that we've defined. So let's walk through that. Here's our simulated cell, which occupies this region of the lattice. And now we're going to say that we're going to pick randomly a pixel in that cell, this red pixel, and we're going to imagine that this cell is trying to move into this neighboring site. The membrane of the cell is going to try to protrude out a little bit. And this is called a pixel copy. And its result would be that the cell would gain 1 unit of volume and the neighboring cell would lose 1 unit of volume. If this copy brings the cell closer to satisfying its constraints, then we're going to do it. If this copy is going to make the cell move farther from its constraints, we might say don't do it at all. And if we did that, we would discover that our system would get stuck. And so in reality, what would happen is that there is in fact energy used to move the membrane of the cell, and that energy can overcome opposing forces from the outside. The cell can push against forces that are acting on it. And so the probability of the cell doing something that's unfavorable to it corresponds to the ability of the cell's cytoskeleton to overcome forces that are opposing the cell's movement. Again, the analogy between the biology and the physics is not perfect, but it's not too bad. The reality is that the physics of cytoskeletal movement is pretty complicated, and we're oversimplifying it. But in fact, cells do move uphill. Unlike water, which flows downhill only, cells are active movement, and they can move uphill. Okay. When we do this move, we can have a situation in which one cell wins by coming closer to satisfying its constraints, and the neighboring cell loses. And so the balance of interactions is going to be determined by the costs for the system as a whole, because you can have your two cells fighting each other. And we can have the cell also, here we have the cell gain 1 unit of volume, red copied onto this neighboring site, green. Here we have red copying on to the neighboring site, so our cell is losing 1 unit of volume. Again, whether we make that choice or not is going to be probabilistic. These are stochastic models. Okay. There's a particular algorithm which we're going to follow that implements this. It's called modified Metropolis dynamics. There's an old, very, very famous paper from the 40s by Metropolis, which invented this methodology. We had to change it a little bit because it has some beautiful features and some features that don't correspond to biology. But there's going to be the same thing, and this is going to be run literally billions of times or hundreds of billions of times in your simulations. We evaluate the current energy of the whole cell lattice. We pick randomly one lattice site in the simulation at a time. We ask the question, we pick a neighbor to that site, one site at a time. We ask the question, what happens to the pattern effective energy as a whole if that site that we picked as a source overwrites the site we picked as a target? What sites we could pick as targets are going to depend on that interaction range that we talked about earlier on, which is why we have to make that decision early. If the source and the target are both in the same cell, nothing happens. Having a volume of cell 1 replaced by a volume of cell 1 does nothing, so we ignore it. If the two voxels we picked belong to different cells, we ask the question, if we made the substitution, what happens to the energy? This is effectively measuring the force that is provided by the universe, either pushing us in the direction of making the movement or opposing it. If the pattern energy goes down, we make the change. If the pattern energy goes up, we roll the dice and we make the change with the probability, and this is physics now, and I'm not going to go into great detail about why we use this particular form. We make the change with a probability that is exponentially decreasing with the energy cost. And that energy cost is rescaled by a parameter called T, which has the very unfortunate name in simulation of temperature. And the reason it has the name temperature is because in the physics that this is derived from, it represented a fluctuation temperature in a particular kind of model called an Ising model. In reality, that T represents the force or energy that is generated by the cytoscalp, which determines the probability of pushing uphill. And so calling it a fluctuation amplitude is more accurate. Once we've made that dynamic decision, we can ask a question, which is, how fast does a boundary move? How fast does an object move? And to do that calculation, and I'm going to skip because we're late. I promised people a break, and we're way past our midpoint of the class. The way we're going to calculate the probability of a boundary moving is by asking a question. Suppose that initially this voxel 2 is going to overwrite voxel 1 here. And I calculate the probability of that happening. I then calculate the probability that at the next time step, I undo the thing that I just did. So I imagine a boundary moving forward and then moving backward. In one case, because they're symmetrical, the energy of one is minus the energy of the other. That means that one of those two energies is always going to give me a probability of one of the thing happening. The other probability, the other direction, will give me a probability of e to the minus the energy divided by t of it happening. The net rate is going to be 1 minus e to the minus the change in energy divided by t. If I assume that the change in energy is on the order of the temperature, the fluctuation amplitude, then I can Taylor expand this exponential, keep the lowest order term, and I discover that the velocity is delta H, the change in energy, divided by the length scale of the lattice, which means that the velocity is equal to the length scale squared divided by the time scale divided by the fluctuation amplitude times the force. And this gives me the physics that I want, because the velocity, the net velocity at which an object moves, is proportional to the force that's applied to it. The drag, which corresponds to mass in Newton's law, is given by the length scale of the lattice squared divided by the time scale divided by the fluctuation amplitude. And so ultimately, I'm given the result that gamma times the velocity is equal to the force. Gamma now is the reciprocal of what I just said, temperature, time scale divided by length scale squared. So velocities are linear in the force. And that corresponds to the reality that applies in the subcellular world. And the stochastic fluctuations correspond to the reality of cytoskeletal fluctuations. And so the result is that if I have some complicated set of constraints, the system will evolve to satisfy those constraints, provided that the force is not too big compared to the fluctuation amplitude. And this is something we're going to do again and again. This is Giulio Belmonte, my old student's slide, explaining this. If the movement forward has an energy with something that reduces the pattern energy, I accept it with probability 1. If the reverse movement has an energy that's opposed to the movement, I accept it with a probability that decreases with the energy cost of the movement. The key scaling factor for this curve, as I reduce my probability, is that T, the fluctuation amplitude, that determines how big an unfavorable event I can accept. Let's look at this in practice. I'm going to show you 2 simulations of a single cell crawling around. And I want you to ask the question, which one of these 2 simulations has a higher fluctuation amplitude? The left one or the right one? Higher fluctuation amplitude means that unfavorable choices are made more frequently.
So the right in that case.
Yeah. You'll notice that the left cell looks like an amoeboid cell, maybe a fibroblast crawling on the substrate. On the right, I see some things that probably I don't want the cell really to do. I see pieces of the cell scattering off and disappearing, the cell splits in pieces, and so on. So in this case, I've got fluctuations that are too big to be biologically realistic. The left, it's reasonable. Here's another pair of simulations with the same question. This is now not a single cell, but this is cell rearrangement. Which one has higher T? Which has more fluctuations? And this is a little bit of a trick question because something else has happened here that makes this not quite what you think it is, perhaps. Remember that what matters is not the fluctuation amplitude per se, but matters is the ratio of the energy changes of the cell compared to the fluctuation amplitude. If you look on the left, you'll notice that all of the cells, blue and green, have surfaces that are pretty similar. There's some fluctuations on the surfaces, but the cells have relatively smooth boundaries. That means that the typical energy scale of the cell and the typical energy scale of the fluctuations are about the same for blue cells and green cells. If I look on the right, the blue cells, the boundaries are completely distorted. The cells are falling apart. That means that the fluctuation amplitude is large compared to the energies of the cells. The green cells, on the other hand, are getting to be very square. They're lining up on the cell lattice, and they're not moving very much. That means that the energies of the green cells are high compared to the fluctuation amplitude. So in the blues, on the left-hand side, the blue cells and the green cells have both been adjusted so that their intrinsic energies are on the same scale as the fluctuations. On the right-hand side, the intrinsic scale fluctuation of energies for the blue cells is too small, and for the green cells is too big. And so the right-hand side illustrates a pathology of the model. The left-hand side illustrates a model that's tuned appropriately. All right. One other thing before we stop for a 5-minute break and then do some coding. The simulation steps consist of lots of micro steps, what are called spin copy or pixel copy attempts. In a pixel copy attempt, I pick one lattice site and one neighboring site. I try to update the neighbor, overwrite the neighbor. I evaluate the change of energy, and I determine by rolling the dice whether I make the change or not. That happens at one point in the simulation at each microstep. Clearly, that time scale isn't so interesting because it takes a long time for anything to happen over the whole pattern if I'm only doing one tiny update at a time. And so the intrinsic time unit of my simulation is the number of microsteps it takes to sample the whole simulation. And so the intrinsic time scale of the simulation is given by the number of lattice sites I have, attempts. So if I'm in 100 by 100 lattice, I will do 10,000 pixel copy attempts. That's one time unit. And that's called the Monte Carlo step. And the reason it's called the Monte Carlo step, again, is physics language. It is these kinds of simulations where you roll the dice to determine whether you make the change or not are called Monte Carlo simulations. I suppose it should be a Las Vegas simulation these days, but Atlantic City simulation. But in any case, it dates when that was done. And so you're going to see in CompuCell that the intrinsic time unit that we use is called a Monte Carlo step, MCS. And in fact, within CompuCell, when you want to know what time you are in the simulation, you don't ask for the time, you ask for MCS. The other thing that I'm going to talk about a little bit is the structure of the simulations, which is that in CompuCell, we are going to alternate time stepping the movement of our cells by a little bit, because one Monte Carlo step is not a long time in terms of cell movement, with other kinds of calculations. We're not going to run them at the same time. We're going to alternate them in time. And so a CompuCell simulation will consist of sweeping the cell lattice with one update attempt worth of updates per lattice site. And then we will stop. We'll leave the lattice configuration alone. We'll do everything else that we want to do. And then we'll do the updates again for the spatial configuration. Because one Monte Carlo step is a step, somebody had the brilliant name of calling everything that we run alternately with the movement a steppable. Another terrible name. And so you will see simple growth steppables. And the reason they're called steppables is because they are stepped in between the movements of the cells. And so that's the basic structure of the simulation. Okay, let's take a 5-minute break, come back at 7:20, and then we'll use our last 40 minutes of class to begin to explore with pictures and simulations the meaning of the mathematics and physics I was talking about. As I said before, if you've had the physics and the math and that was meaningful, that's wonderful. If it isn't, I hope that when we see how these things play out in a simulation, it'll make more sense. So I'll see. Are people ready to get our hands dirty a little bit? Now, certainly hope be less mathematical for the rest of the class. And I promise to the biologists in the room that typically we have very little mathematics. But today we're trying to get as much of the mathematics out of our way so we don't have to think about it so much going forward. So we're going to do a little simulation here that Giulio created, which is a sort of a toy representation of what he thinks of as an epithelial versus mesenchymal transition. I'm not sure I'm convinced about that, but it has some nice patterns. For those of you who are not biologists, we are going to think about two situations, one of which, and this is shown from the side rather than the top, in which the cells are going to form a compact layer, like the tiles in a bathroom tile floor, versus a situation in which the cells are spread out with some space between them and a little bit more disorganized. And here I'm going to skip a set of 10 or 12 slides which go through the details of what the difference between epithelial and mesenchymal cells are. And if you are unfamiliar with that, I encourage you to look at the slide deck. I've left the slides in place, but they're hidden. And so now I'd like us to work together and to build a little simulation of this. And I'm sorry, people said they'd preferred to have things, to have the slides full screen, but I'm going to have to go back for the moment to the to the split screen. So I'd ask you to go into Twit, and I'll give people a minute to get Twit running again. So go into Twit, and let me get rid of this for the moment so that we don't have all so much on the screen at once. I'd like you to go into Twit, and we'll do the same thing we did last week, which is we're going to create a new simulation. And so go up to CC3D project, new CC3D project. And then give people a few seconds to get Twit launched. And make sure people can follow along. Okay. And so we could give this a name. Giulio gave it a name that was meaningful in the context that he gave it, but we could call it whatever we like. I'll call it EMT for epithelial to mesenchymal transition. At least I will if it'll let me type EMT. And we'll leave out the defaults otherwise the same. Make sure that the simulation directory we're pointing to has your name in it, so it's a simulation directory you can write to. Hit next. And we can leave everything, I think, as is in this simulation. Notice this pixel copy range, which is that interaction range that I was explaining earlier. So that number now maybe has some meaning as we've seen it in the simulation explanation. Okay, hit continue or next. And we're now going to create a single cell type. He says to call it my cell, so let's call it my cell. We type that into this line here, my cell, hit add. Next. We're not going to create any chemical fields, so we'll skip that, hit next. And now this is the window that we're going to use so much in specifying simulations, because this is specifying now the plugins that specify those constraints and energy functionals that we were talking about. We're going to give the cells a contact energy. We're going to give them a volume. And that's it for the moment. And we're going to hit next. And we're now done with the simulation, and we'll hit finish. And you'll notice that Giulio's used a Mac, and so some of the menus have a slightly different appearance, and even some of the dialog boxes have slightly different wording. But functionally, they're the same. Was everybody able to get through that? Or do people need me to do, I could walk through it again if people would like to go back through the steps. Does anybody need to review any of the steps, or were you able to get through it? Again, I'll walk back through it quickly. New CC3D project. Name of the project. No changes to the simulation properties. Create one cell type. Hit add. Continue. No chemical field. Select contact. Volume flex. Center of mass. Continue. Done. All right. Okay, so we now have our simulation here. We go right click, open in player. And in nanoHUB, we'll have to select the window here and resize it. You can use that tile function to display better, and you should see the simulation running. And it's a little bit hard to see with this scale, so I'll zoom in a bit, and you see the cells. You'll notice these cells are fluctuating a little bit. They're not square, so they're not lined up with the lattice, which means that the energies are picked about right compared to the fluctuation amplitude. And this is the basic simulation that we're going to work with. And let me just check to make sure that people were able to get that simulation to work. If anybody needs help with this, let me know. Giuliano is also here to help. So you're free to, if we need to, we could always set up a breakout room and have, or if you're having trouble, probably everyone's having trouble, so we're happy to go over this. If you are caught up with this and want to try playing with it, you can try changing the imaging by changing the colors under preferences here. You can figure out how you can screen scroll and become familiar with using player. There was one person who said they needed a little more time. Don't want to rush you, so just tell me when you're ready. So this, in Giulia's view, is something that looks a little bit like an epithelial sheet looking from the top. The cells are all stuck together. They're compact. They're columnar. And so this is a simple model of something that looks like an epithelial layer. And you'll notice that there are random fluctuations, and each time you run it, you're going to get a slightly different result. If you ran exactly the same simulation 20 times, it would be similar in its bulk features, but the exact position of the cells would be different each time. Okay. If we stop this simulation and restart it pushing the step function, so we see the initial configuration. Let me get it started again. Let's see. Stop. All right, CompuCell crashed on me. Just to show that I'm not, if it's crashing, it's not you. It's, okay, let's try again. Actually, the simulation is saved, so I can just open it. Okay. So what I wanted to do was show you the initial configuration. Initially, we start out with a square grid of cells that are all identical. And you'll notice that they're lined up on the cell lattice. And now I'm going to do a single time step. And you'll notice that randomly the boundaries move. And they move by 1 pixel. Because the most we could move in one Monte Carlo step is 1 pixel. If I keep time stepping, you'll see little notches will be made in the boundaries. Occasionally, you'll see a boundary move forward and then move back in the same place, which is what we discussed before. As time goes on, that initial square symmetry, which was not biological, will disappear, and the cells will begin to take on more rounded, relaxed shapes. You can see here is a cell that's pretty round. Here's a cell that's pretty round. On the other hand, if I look in the upper right-hand section here, all the cells are pretty square. So it takes some time for the simulation to gradually lose its memory of that square initial condition and relax to something that looks more reasonable. This is exactly what we were talking about the probabilistic, stochastic relaxation of the pattern to reach a state that corresponds to the optimum under the constraints that we've specified. If I zoom in, you can see that there are some fluctuations, but the basic geometry doesn't change too much. If I zoom out, the shape of the whole tissue is still pretty square because I started out with a square, but if I run this long enough, it'll gradually round up and make something that's more or less circular. Okay, is everybody getting that? The main issue here is that all of this dynamics is happening primarily because of this contact energy that we've specified. We've talked about constraints. The last concept that we're going to introduce today is going to be contact energy. And the contact energy conceptually is very simple. If I have two objects and I bring them into contact with each other, there is some energy associated with that contact. If they repel each other, that energy is positive. I gain energy by moving them apart. If they stick to each other, I have to apply energy to bring them, to separate them. That means the energy of contact is negative. And that sign is a little bit confusing, perhaps, to begin with. If things stick together, their energy is negative. If things repel, their energy is positive. Because in physics, you're always trying to reduce energy. So you roll downhill. If the energy is negative, you roll towards it. If the energy is positive, you roll away from it. And I have to define the contact energy per unit area. And this is where that lattice range comes in again, because the surface area depends on the lattice I'm using, how many neighbors I have. But conceptually, this is simply the surface energy. If I have a free surface, I have an energy per unit area between the cell and the medium around the cells. If I have two cells contacting each other, I have an energy per unit area between the two cells. Biologically, if that black region were extracellular matrix, the adhesion between the cells and the ECM would be dominated primarily by integrins. The more integrins I have, the more I stick to the extracellular matrix. There are certainly kinds of extracellular matrices that would repel the cells, and that would give you a positive energy. The cells would stick to each other through things like cadherins. The more cadherins the cells stick more tightly to each other. On the other hand, there are molecule species like epinephrine, which cause cells to repel each other. So if the cells had F-ephrine pairs, there'd be a positive energy that would push the cells apart. So that's the biological explanation for the same thing we're seeing physically here. In this case, when we specify contact energy, for each pair of cell types that we have, and here we only have one basic cell type in medium, so we have two possible energies, I have to specify the energy per unit area between that pair of cell types. The trick is that when I'm doing a sum over the contact, I have to sum over the range of interaction. Nearest neighbor one is pretty easy. When you go to third neighbor or fourth neighbor, it's harder to keep track. Fortunately, the computer does that for you. And if I want to write it mathematically, it's a sum over all voxels in the simulation of a sum over all the neighboring voxels of the energy per unit contact times the type, for the types of each cell, provided that the voxels belong to different cells. In our case, we only have two cell types, the medium, the black here, and the cells that we defined. And so we only have three possible contacts. Medium to medium, we only have one medium cell, so we never have medium touching medium. Cell contacting cell, and medium contacting cell. So we really only have two energies to worry about. All right. So let's see what happens when we change the contact energies between cells and cells and cells and medium. Let's go back to TwidIt. Let's open our simulation. And let me see if I can, I'm going to expand this a little bit here. Everything we're going to do now is going to be in our XML here. So select the XML. Now let me see. Okay, can everybody see the code now? So I'd like you to follow along here. So again, did everybody have the code running? Go back to TwidIt, double click here at the top where it says the name of the simulation. Select the XML here, emt.xml. You may have to double click on it. And then scroll down, and it should be about line 36 or line 37. So again, double click on the name here, double click on the XML, and then scroll down. Again, it should be line 36 in your simulation. Everybody got that one? Yeah. Okay. We see something called plugin, name equals contact. That's saying that the cells have a contact energy with each other. And then we're going to specify this energy between a cell of type medium and a cell of type medium. That's not going to do anything. Between a cell of type medium and a cell of type micelle, a cell of type micelle and a cell of type micelle. And it says neighbor order, and this is the range over which we count. And let's now change the medium micelle contact energy from 10 to 20. That's actually making the medium repel the cells more because a positive energy is repulsion. So you say medium micelle 20, And then I hit save, this double floppy disk up here, save. You can also do a pull down here on the file menu and select save all. And if I change this and don't save it, you'll notice it's a little bit small, but this icon here becomes red. So if I do the save, that icon will become blue. So if I look carefully in the tabs at the top, it tells me whether I've saved my simulation or not. And now, in principle, if things are working, player will know that I've made a change. So if I stop the simulation and now start it again, it should load the new simulation. Has everybody got that to work? And you'll notice it's different. Exactly how it's different might take some time to think about, but it is different. Notice it's rounding up here on the boundary between the cells and the medium much faster than it was before. And if I zoom in, the fluctuations between the cells are a little bit less. That's because the whole system is under compression. Okay. If I want to see whether the change was made or not, and this may be a little bit hard to see in the reduced window. Within Player, you see this box that's called Model Editor. It's this list here on the left. This allows you to change parameters in the simulation while it's running. The editing that we did in Twidit changes the values for the next simulation I run. I can change parameters in the simulation while it's running using this model editor. And I want to do that together. If I look in this list of plugins, I see cell type, volume, center of mass, contact, uniform initializer. I'm going to open the contact plugin by clicking on that arrow. Go ahead, Connor.
When you change the parameters in that section, does it overwrite the original code, or is it just for that instance of the simulation?
It's just for the instance of the simulation. And something that I've wanted to have added to CompuCell for 10 years now that nobody gets around to is that you should log it. Because at the moment, it doesn't keep track of the changes you made by hand. And so that means that if you find, gee, it was really neat when I did this, and you didn't write it down, it's lost. And so there really should be a file that keeps track of every change that's made in this model editor. It says, at time such and such, the user typed such and such. But we don't have that. That's a good question. The other little inconvenience that you're going to find is that when I open the plugin, I see the numbers here on the right-hand side, but I just see energy, energy, energy. And if I want to see which energy it is, I have to click and open that next hierarchy. So if I click here, it'll say medium, medium, contact energy is 10. Here, energy type 1, type 2, medium, my cell energy is 20. It's not so bad when you have 2 cell types, but if I had 36 cell types, I'd have 36 times 36 values. And to have to scroll through all of those, opening them up to see which is a pain. So another thing that would be nicer would be if it said energy and then gave you the name of the energy there, rather than making you do it by hand. However, for our purposes, it's okay. So we're going to open up medium micelle, and we can open up micelle too. And we're going to change that medium micelle energy. And so why don't we put back that number we changed from 20 in the simulation specification, we'll change it back to 10. Click here, type, and I have to hit enter. And there's a minor warning, which is if this was a decimal, you have to have it be a decimal. If it was an integer, you have to make it an integer. And this has to do with the way XML processes tags. So if you type 10 with no decimal point, it won't work. You can't see a huge change here, probably. But you can see some change. And if I want to see what that change was, I could go back to 20 and see that the surface rounds up. See, the surface here is much smoother. If I make it 10 again, the surface will get a little bit rough. Maybe I should zoom in a little bit. Here's what I get for 10. Here's what I get for 20. You see this fluctuations reduce? And so this is showing that smoother simulation. And if I compare it, it's a pretty dramatic difference. The bigger the energy, the stronger the force. I'm basically pushing in on my tissue more when I make that energy bigger. If I make that energy too big, the boundary will line up with the lattice, which I'm seeing here. And the system will become octagonal or square because of the lattice symmetries, which I don't want. Now, why don't you try making it 5 instead of 20? I'm not going to do that. I'm going to do it, but I don't want to. I want you to tell me. Somebody tell me what happens when you make it 5. Anybody? Joel, go ahead.
It becomes more jagged on the edges. It almost like the cells wants to spread apart. You're talking about cell and medium, right?
Yeah. Yep, you see the medium is beginning to penetrate between the cells. The cells aren't really sticking very much to each other. Why is that? Remember, positive energy is bad from the point of view of the system. So the cost of the cell medium energy boundary has gone down. So we get something that looks like that. It's only at the surface because it's only where cell and medium are touching each other that it makes a difference. Okay, so why don't we now do the last step? Go from 5 to 3. What happens?
Before you said that, I went to 0, so I went a little bit too extreme. But it starts to stop containing itself as we get lower. Yeah, there.
The cells crawl off. They stop sticking to each other and start crawling out into the medium. And so this is our tiny simulation of epithelial to mesenchymal transition. We go from a situation in which the cells are tightly adhering to each other and don't want to stick to the medium around them, to a situation where the cells don't adhere to each other and do stick to the medium. And so this is a reasonably simple simulation of something that does happen biologically. It happens functionally in early development. It happens pathologically in cancer, where cells begin to detach from the primary tumor and migrate out. If you put this number back to 10, what happens? Anybody?
The cells start to reform.
The cells are going to start clumping together again. Exactly. Now it takes time because here they're less motile, so it's going to take quite a while for them to find each other. But that kind of distribution and recoalescence does happen in development, particularly in early vascular development. Peripheral vasculature happens like that. There are other situations in which that happens too. If I waited a long enough amount of time, these would all recoalesce into one big blob. It takes a long time. So we can get the fragmentation, and if we put the energy back, we'll get the clumping again. Okay, we're out of time, so we didn't get through everything. I would ask people, it will probably take me till tomorrow. I will assign the remainder of this exercise, which is to explore what happens. So we can do, well, let's do that last thing. What if we changed the cell-cell energy and made it smaller? Let me zoom in so we can see a situation. Let me make the cell-cell energy smaller. Let's make it 5. What happens if I do that? Actually, he says 1, so let's make it 1. We'll make the cell-cell energy 1. If the cell-cell energy is 1, the boundary has a low cost, so the boundaries between the cells fluctuate a lot. They get rough and they fluctuate a lot. Notice how round these droplets of cells have gotten. Before they were rather pinned out. Now I get these nice circular blobs of cells, and look at these two blobs coalesce. There's another blob that's coalesced. So this is actually seen a lot in cell adhesion and cell sorting in early development. So the smaller the energy of contact compared to the fluctuations, the more fluctuation we get, the faster things move, and the smoother the boundaries are going to be with respect to the outside world. So I think that that's a good place to stop. I'd encourage you to keep playing with this. There are actually quite a few situations you can play with because you have really 3 numbers. You have the cell-cell contact energy, you have the cell medium contact energy, and the relative values matter, but also the ratio of that fluctuation amplitude. We haven't changed the fluctuation amplitude. So the ratio of cell-cell to cell medium energy matters, but also the absolute scale matters. Okay. Are there any questions before we stop for tonight? The one thing that I wanted to do was look back at the homework, because as I mentioned, we might or might not get through all of it. Problem 1, we can do. That's taking the simple equations that I gave you in class and calculating it by hand for one cell. Problem B, we can do, same thing. Problem C is basically exploring the simulation that I've done. 3 is exploring the simulation that I've done in class now. So 2 and 3 we can do. Problem 4 we can do as well, which is changing the target and the surface, although we'll come back to that in class. And I gave a little extra credit for people who are mathematically inclined, where you can explore the math of what's going on in these simulations. The one thing that we did not do, which would apply to problem let's see, did I ask for it or not? No, it's okay. I don't think there's anything in here that we didn't cover. But if you run into something where in the homework where you say we didn't get to it, that's all right. Say so. Okay. Any final questions before we break? Again, I know this was a little mathy today, and I'll try to. There will be some math going forward, but this will probably be the mathiest class of the series. The other place we'll run into some mathematics is when we talk about the diffusion equation, because there's really no way to talk about chemical diffusion without doing a little bit of math. And maybe when we do chemotaxis, there'll be a little bit. But mostly the math is in how surface energies and constraints work. And again, knowing the background mathematics may be helpful to people, but fundamentally you can treat this as an experiment where you change the surface energies and you see what happens. You change the fluctuation amplitude and you see what happens. And so one of the things that this whole simulation environment is trying to do is to make it easy to do those kind of experiments, those kind of computational experiments. Okay. So, in that, I think people are probably hungry. It's 8 o'clock, and I hope people get a decent dinner, and that we are able to go forward with this. And I look forward to talking with people next week. And if people, the rest of this week is a little busy for me, but if people would like to meet to talk about projects over the weekend, I can set up meetings over the weekend if people like to do that. So you don't have to do it this week, but and people have met each other. So if there are possible shared interests, I encourage you to reach out to each other directly to talk. Okay. Thanks, everybody. Appreciate it. And I look forward to seeing you next week then. Good night.
Night.
Thank you. Good night.